in mitochondria regarding to acetylation is unique. Why it's unique? Because any other location in the cell, acetylation is tightly regulated. You have enzymes that add the acetylation and enzymes that remove the acetylation.
Welcome to Longevity by Design, a podcast designed to give individuals access to the leading scientific information in the field of longevity. The ability to add years to your life and life to your years needs no opinion. Join us as we ask science to take the wheel. In each episode, Dr. Gil Blander joins a co-host and an industry expert in the field of longevity, shining a light and getting the answers to the key question: how can we live a longer, healthier life?
Hello, I'm Dr. Gil Blander. Welcome to Longevity by Design, how to live a longer, healthier life. We are produced by Inside Tracker, your science-based guide to optimize your body from the inside out. Today, we welcome Dr. Haim Cohen, a professor at Bar-Ilan University and a global leader in aging biology. Dr. Cohen is the founder-director of both Minerva Israel Germany Center for Biology and Mechanism of Aging and the Sagol Center for Human Health and Longevity. Dr. Cohen has authored 75 peer-reviewed journal articles, most notably on the role of the SIRT6 enzyme in extending lifespan by up to 30% in mouse models. His research span multi-omics, metabolomic, and translational intervention, including the SIRT6-driven modulation of glucose metabolism and inflammation. Welcome, Chaim.
Hi, Gil. How are you?
I'm very well. So, Chaim, we interviewed you a couple of years ago, and we discussed your exciting paper about SIRT6 role in longevity. Can you please provide us with an update where this study is standing, any follow-up experiment that you have done? What is exciting that you have in the oven right now that you can tell us about it?
Which excitement do I have about 36? So actually, still, let's say half of my lab is still doing 36 biology. The other half moved to something which I will describe soon. And mostly what we're doing now, we're focusing on tissue-specific 36 effect. And we do see that some tissues, although it's not published yet, some tissue, if you overexpress 36, you get similar effect, not exactly the same. Here's whole body 36 overexpression, which is by itself is exciting. So now we can focus on one tissue instead of research which part of the body is important. We also did a lot of work about 36 role in the brain. This is something that we're writing now, and so we saw that 36 in all the animals actually allows them to have a metabolic balance in the hypothalamus, which is the part of the brain which is important for your metabolism. They have the same energy status, or they restore the energy status of the hypothalamus like in young animals. The result is that the hypothalamus response to exercise maintained as in young animal, and by this, they have the same activity like young animal. Because what we saw before was that when you overexpress 36, they have a similar activity like young animal, but actually we didn't know exactly why. So what we did now, we focus on the muscle. We didn't finish it yet, and we did extremely huge experiment of multi-omics like metabolomics, all the omics that you can imagine in the hypothalamus to see what's happened when you have overexpression of 36 in the hypothalamus. And we found that 36, when you overexpress it in old age, it maintains the energy of the hypothalamus, the energy pathways in the hypothalamus, and by this regulate other tissue that you have similar activity like in young animals. The last project that we have now is we're following the role of 36 in regulating chromatin structure, global chromatin structure. Not like, when you're young, or let's say differently. 36 is deacetylase, meaning that it's remove acetylation, and by this, you control transcription because the chromatin is open and closed. But this is in a single localization. What we did now, we followed the global effect. Apparently, as we age, the chromatin in many sites is open instead of maintained closed. In some sites, it's the opposite. So it used to be closed, now it's open. So it used to be open, now it's closed. The outcome of this, we focus on the liver, is that areas that used to be open, meaning this area was responsible for the expression of tissue-specific genes, now become closed, And because of this, the tissue losing their identity. So it used to be a liver when you're a young animal or young human. Now when you're old, you don't express well the liver genes, but on the other hand, you express other genes. And if we are now focusing on the architecture of the whole chromatin to see whether 36 is involved. This is what's happened to your chromatin structure as you age. This is something that we came out of the main avenue of 36.
Yeah,
So
no, that's exciting and sounds like the data that we discussed a couple of years ago is still valid, and looks like you're even trying to understand the mechanism, what issue. Is it still gender specific, so more affecting one gender than each other, or you don't know yet?
Oh, it's extremely gender specific. That's, and I wish that I knew why gender specific. That is the biggest question. I think in aging, in many pathways in aging, you see gender specific effect. Like if you think about mTOR, the downstream of mTOR, the S6 kinase, it's more expressed in female, for example, the effect of mTOR. The effect of S6 kinase, so which is downstream of mTOR. In 36, we saw that it's mainly, we have double effect in male. Now, when we do it in tissue specific, in adipose tissue, for example, again, it's gender specific. This is, and I would like to say, unfortunately, because it's making our life more complex, but we have target of 36, which I'm not going to explore now. I hope that we can explore it soon, that we took the acetylation site of a protein, which is not a chromatin, and 36 deacetylated. We create by CRISPR constitutively acetylation site and constitutively deacetylation site, and still gender specific. Even though that we just took it to downstream to the one target out of, I don't know, dozen of 36 target, and we still see gender specific effect. Yeah. I think it's a big question in aging field, why some of the effects are gender specific, and hopefully we have enough funding, because I know what is going on in the US and all across the world, that the NIH and other institutes will invest in investigating why there is, probably they will, but to investigate what is the source of the gender specific effect.
No, it's definitely very interesting, and thank you so much for double click into the 36 story. But today we decided to meet again, not because of the 36, but because of a very exciting paper that you published, I would say a couple of months ago, in the scientific journal Nature, which is, for our audience, if you don't know, it's let's say one of the top 3 journals in the world, at least in the biology science. And in that paper, what you showed, or what you published, is basically you look at a global effect of acetylation in different, if I recall correctly, it's 107 different mammalian species. And you show some interesting result, but before we dive into the paper, can you just explain what is acetylation?
Okay, acetylation, you can imagine that you have a big machine. This can be a protein, this can be us. And within this big machine, you have the on and off switch. It's very simple like this. So think about a protein that his weight is 70 in the unit of protein. In human being, let's say that you weigh 70 kilograms, and they just add to you a very small modification, very small change, which is 40 grams. This is the difference between the whole protein and the acetylation site. And by this, I completely change your activity. I think you're very familiar. I know that you're familiar, but the audience probably don't know. But in P53, one of the proteins that Gil worked in the past, this is one of the main proteins that is the first protein that people show that acetylation make it on and off. People say it can be on, it can be off. So but you have dozens of them. Actually, you have tens of thousands of this. There's tens of thousands different on and off switches.
So it's in a way, if I will try to repeat what you said, it's in a way a post-translation modification of the protein that basically in the acetylation you add acetyl group to amino acid called lysine, And when you add or remove the acetyl group, it can change the conformation of the protein or the activity of the protein, and by that regulate its activity. That's fair to say?
Exactly. It can change everything. It can change
Yeah.
with which proteins will interact. It can change the localization of the protein,
Yeah.
can make it active, not active, everything.
Okay, good. So now a follow-up question. Today, at least in a chromatin modification, everyone is looking at methylation. So why have you decided to look at acetylation and not methylation?
Okay. Methylation, why? First, I think the simple answer, well, it's divided too. Okay, maybe I should, later I will explain the rationale why we did it, but third fix is deacetylase. So, and we have, we did in a different project, we try to characterize all the acetylation sites that third-fix regulate. So this is one of the reasons why we took this project, because we started to create a database of acetylation. There is no good database of methylation. The methylation that people look at, it's methylation of the chrome mostly on the DNA. We looked at acetylation on the whole protein, on the whole protein, which meaning within 28,000 protein, hundreds of thousands sites of acetylation. And we know a lot about acetylation. This was one reason why we decided to focus on acetylation. Also because we know that acetylation is tightly connected to aging because of our work and other people's work that found change in acetylation that regulate aging.
Okay, sounds good. Makes sense. So now, Chaim, after a lot of introduction, can you walk us through the design of the experiment, the platform that you built, and explain to our listener what was the goal of this study?
Okay, so we start with I will give two short stories that will explain why we focus on it. The first is a simple event in my lab. Like I used to be what we call a wet lab, meaning that we do an experiment on the bench and we're very far from computer studies. Right? It's something that if you're familiar with different labs, they have the wet side and you have the computation side. It happens that third of my group become computational biologists. And this by itself means that we need to find projects which fit to computational biologists. They cannot build transgenic mice. They don't have the experience for that. They can do extremely imaginary experiment, but not something which is dealing with reagents and so on. This is the beginning of the story. The second part is that we thought, as I said before, acetylation, or let's say any post-translational modification, actually it's helping you to respond to the environment. For example, you eat, and let's think about other modification called phosphorylation. So you eat, and now all the signaling from having sugar in your blood to uptake of glucose and going to growth and the differentiation, or this is coming by phosphorylation of different protein in the signaling. You're under stress. You have different type of acetylation that you need to respond to the stress. It's cold. You need to activate the burning of brown adipose tissue. Everything that we do in the response to the environment, it depends of many post-translational modification. And the ability of us to respond to the environment is decline as we age. I think we all know it. So we thought that maybe if our ability to respond to the environment is decline, and this is mediated by post-translational modification, all we need to do is to find which post-translational modification mediate the decline in the response to the environment. It's a very nice question. After 5 minutes of thinking, I realized this is a dream. Why it's a dream? Because we have, let's speak about acetylation, but we also did, and we have a paper that we're writing now about phosphorylation. In acetylation, in human, we map about 23,000 sites of acetylation. In mouse, we have 28,000. It's not that they have more, it's just we map more. In phosphorylation, we have a database of almost 500,000 phosphorylation sites in the human protein. Protein, it's about, we map something about 150,000 in mouse protein. So I can't come and just do mutation and change its site and see what will happen to the lifespan of the mice. It will never end. I will nobody will come to my lab, nobody takes this project, and it will take me hundreds of years to finish it. So then I realized that probably we don't really need to do it on the bench. We can do it in silico, meaning all we need to find is a lab, which is a big lab, enough lab, that already did it, meaning that they have mutations that mimic constitutively acetylation or constitutively deacetylation. And we have this lab, it's called evolution, because during evolution, some acetylation site, and this now come to more scientific part of it, if you have a lysine that is acetylated, it look like glutamine. If it's deacetylated, it look like arginine. So what we need is to search in evolution which site was converted either to glutamine or to arginine, and it happened only in long-lived animals, or vice versa. It used to be glutamine or arginine, it's converted to acetylated lysine. And this is why...
So Haim, one clarification point that I want to provide to our listener, because again, it might be, as you said, technical. So basically, there are 3 amino acids that are pretty similar: arginine, glutamine, and lysine. And what is interesting, and as you said, that's the lab that you found basically in nature, that you can use those 3 amino acids basically to look at a mimetic of acetylation or not acetylation of a specific place, because those 3 playing that role. And then you can look at that and basically come say, hey, this one is actually mimic acetylation, this one mimic deacetylation. And then I assume you looked at the 107 different animal from the mammalian kingdom, and basically try to see how those location, if you look at specific protein and try to find the similarity in other organism, how are they looking? Are they looking as acetylated, not acetylated, or can be acetylated, correct?
Exactly. That's exactly what the student did. So I came back to the computation part of the lab, and I gave them what Sarit is the student who did it. She created the tools they created several databases, which is first, the proteome of 107 mammalians. Second, the acetylation of long-lived animals and short-lived animals. In other words, human and mouse. And last, the maximum lifespan database. Because she needs now to compare it to maximum lifespan. And what the tool is doing 2 things. The first thing that it's doing is ask which site change only in long-lived animals, meaning that is associated with longevity. Second, she needs to do something which is quite sophisticated, the statistical change, because if you think about evolution, then if you're familiar with the evolutionary tree, actually we and the monkeys are on the same branch. So if we have the same change and we are on the same branch, it's not really exciting, because this could be just because other reason. But if we and whales, which are very far on the evolutionary tree, have the same change, this means it's related to longevity, not to something which is other reason in doing evolution.
It might, correct? You don't have a proof. It's correlated. It's correlated with longevity. Yeah.
That's why they call it, the paper is called the longevity associated acetylation.
Okay. Yeah. No, that's great. And I think that's a very good explanation for our listeners to understand that a scientist spend a lot of time of ideation and finding a good idea to research. And basically, in my opinion, and for my time at the lab, that was the most exciting time, because then you just execute. You spend a few months of thinking about the experiment, and then you execute, I don't know, for a few years. And I think that what you have done is very elegant in a way to come and say, okay, let's look at all the mammalian kingdom and try to see what are the changes in the acetylation site that are similar between long-lived and different between the long-lived and the short-lived. Now, a follow-up question to that, Haim, is what was the borderline? Like you said, long and short, but it's a prism. It's not black and white. You have a lot of gray between. So how have you decided what is the borderline of the long versus the
okay. We didn't decide what is long. It's just a numerical parameter. So you can sometimes see that some of the change happened also in what we would like to call short-lived animals. But so it's not always that it's happened only in a long-lived animal, but we did two-step of statistic to be sure this is significant, so it's unique only to long, what we call long. Usually it's around 25 years of
Okay, but what you said, it's not like there is a borderline. It's more like based on the statistical analysis, you've seen that it's enriched in a longer lived or enriched in a shorter lived, and that's because of the statistic. That's the makes a lot of sense. And
just answer about this. Take the rodents. You can't decide in the rodents. The longest lived rodent should be the naked mole rat, right? But this is 30 years old. So you might say this is not the right borderline, but actually within the rodent, it's definitely it's a good borderline, because within the rodent, this is 30 years old.
Yeah.
And the other live up to 2.5, 3, 4 years maximum.
And the naked mole rat stayed with the long-lived or in the short-lived?
Within all the sites that we found, actually there are very few, I mean, around 11 to 14 sites, which is also exist in what we call long-lived animal and naked mole rat. Naked mole rat is unique. I think it's because of the conditions that they live.
Yeah. And for the audience, we had an episode that we discussed a lot about this animal with Vera Gordenova. So if you are interested, it's a fascinating creature that live maybe almost 10x more than the mice, correct? Around 30 years versus 3 years, which is amazing. But they have more like community, and they live under the ground. And so I agree with you. It's a bit, maybe a bit of an outlier in a way, but a very interesting example of how the same size of animal can live longer, because there is the theory of the size. You are getting bigger, you live roughly longer than much smaller mammals. So yeah, a great explanation. Thank you so much for that. So how many acetylation sites you found roughly that are significantly different between long-lived and short-lived? What is the magnitude of that?
The magnitude is about, depend which direction we look, but it's around a couple of hundred maximum. Out of tens of thousands acetylation sites, at the end, it's come out to around, let's say, 400 different acetylation sites.
Okay, so there are some acetylation sites that enrich in the long-lived and some that are in the short-lived, but it's a very small sample out of the universe of potential acetylation sites.
Right, more than that. There are many sites of acetylation, or lysine. We compare lysine to different amino acids. Lysine by itself is more conserved compared to other amino acids, especially acetylated lysine. So to find something that was changed during evolution, it's more difficult in this group.
Yeah.
As a Longevity by Design podcast listener, you understand the value of improving your health for today and for all the years ahead. And if you want to live your healthiest, longest life possible, you need to understand what's going on inside. At Inside Tracker, we take a personalized approach to health span optimization that eliminates guesswork from your wellness plan. Inside Tracker analyzes blood biomarker and DNA data, along with physiomarker data from fitness trackers like Oura Ring, to deliver personalized food, supplement, lifestyle, and exercise recommendations that allow you to take control and improve your health span. And for a limited time, Longevity by Design listeners can get 20% off at the Inside Tracker store. So if you're ready to receive a personal health analysis and data-driven wellness plan to optimize your body for the long haul, then it's time to start inside. Visit insidetracker.com/podcast to get started today. That's insidetracker.com/podcast to get started today.
And why do you think that it's acetylation, or maybe the other question is, if you will do the same experiment, and you said that you are doing it for phosphorylation, but let's say for methylation or any other glycosylation, would you imagine that you will see similar result, or it's unique to acetylation somehow?
The magnitude of order between the number of sites and the number of sites that you finish the experiment, it's about the same. I think in phosphorylation, it's even less than what we see in acetylation. If I think now in the number, in phosphorylation, it was about 1,000 sites plus minus. We did even more strict because of so many phosphorylation option, because they have different amino acids. We did the FDR that we use was even higher, and the FDR is the statistic for the audience. And at the end, it's finished between, let's say, 1,500 1,500 phosphorylation out of 500,000. So it's even less than what we see in acetylation. So at the end, it's about the same magnitude. I'm not sure what will happen in other modifications, simply because there is no good database for other modification. Acetylation is something that we and other map it for a very long time. Phosphorylation, even many more labs than us did it. So we have a big and solid database. We're doing the ubiquitination, and this is something that we need to build one by one. It's very difficult. We're doing the methylation. Again, methylation, it's a very small group of protein. It's not because it's not exist. I think it's just because the database is very limited. The database is limited, so it's a bit difficult to say anything about this modification, at least now.
So is it fair for me to say that most likely we will see similar result with other post-translation modification? And what is maybe one of the interesting point of this paper is to say that maybe some of the difference between the longevity of different kind of mammals is because of post-translation modification. And right now you came with the light on the acetylation. That's why you see that. But there will be more effect with other post-translation modification.
Definitely. And the next challenge will be how to create, and we don't know how to, and look, that's it. The next challenge we're going to face, one of the next challenges is what is the minimal combination that you need in order to get the best effect? Also between different modifications, like do we need only 5 phosphorylation, 10 methylation, and 7 acetylation, and to find this combination. And what at least my knowledge in artificial intelligence and in machine learning is limited, but I understand the tools that we have now, at least for now, to find this combination are limited because of the small number of mammalians that we have. We have millions of modification, only 100 mammalian. And the existing tools doesn't give you the way to solve this issue. We're trying to find it, but it's look like a very difficult topic in machine learning.
So the hand number of the mammalian species is not wide enough to allow us to find the strong statistic of difference between them when you look at combination. Makes sense. And now we are getting back to an interview that I've done with Ran Sigal a few months ago, and we discussed about how can we use machine learning, LLM, and all of that for longevity. So again, if anyone from the audience is interested, that was a fascinating discussion. Haim, I want to move forward and discuss some of the interesting things that you found. And one of them was that you found that there is basically acetylation of a protein called CBS, specifically K386-2R. And that's a protein that work on the hydrogen's sulfide production. And it's also a pro-longevity a protein in C. elegans, and you validate it in this paper. Why do you think that acetylation of this protein at this specific location is important?
Okay, so let's start with the previous papers that published about 7 years ago. There was a set of papers that were published showing that H2S, this gas that you spoke about, is important to mediate the effect of calorie restriction. There was a set of papers that were published about it showing that H2S production mediate the positive effect of a calorie restriction. And when we looked on the different pathways that we found to be regulated by acetylation, longevity associated acetylation site, we found that one of the main pathways was what's called the 1-carbon or the transsulfuration, which is the pathways that create H2S. So that's why we decided to see what's happened between human and mouse regarding specific to this site. And what we did in the validation, we took the mouse site, converted to human, and we found that now the mouse protein have a better H2S production, which might explain why humans live longer or why mice live shorter, because when we took the human protein, we changed the site back to the mouse amino acid. It's reduced the activity of the enzyme. It's a lower H2S production. What we did next, which hopefully will come out, let's say in 2 years from now, we went back to the lab and create mouse which have this specific modification. Like we took the site and we changed it from mouse to human on this single location by CRISPR, and now we're waiting to see what's happened to the lice.
Okay, so you made a transgenic mice that has the human acetylation site in the mouse of that protein, and now you are looking at the lifespan of these mice?
Lifespan and all the other phenotypes which are related to longevity.
So in this specific example, the difference, or the only difference in this protein is this lysine, or there are other differences that might explain the difference in activity?
We change only specific lysine. We took the lysine, in mice it's lysine, we change to arginine as we see in human, and we change only that.
Okay, so it's literally just that change. Very interesting, and I'm curious now to see the result, as you said, a couple of years from now. That's very interesting, and it is definitely an elegant story. You also, in the paper, you also discuss some a mitochondrial pathway enrichment of acetylation site. Can you elaborate on that? What is the effect of?
Okay. So we don't focus specifically on the mitochondria in this case, but in mitochondria, regarding to acetylation is unique. Why it's unique? Because any other location in the cell, acetylation is tightly regulated. You have enzymes that add the acetylation and enzymes that remove the acetylation. In other words, you have acetyltransferase and deacetylase. In the mitochondria, the regulation is half, meaning that level of acetylation is depend on the activity of the mitochondria more than, let's say, it's depend on the level of acetyl-CoA, and the level of the process of deacetylation is specific enzymes that remove the acetylation. So this is tightly regulated, connected to your diet, because acetyl-CoA is connected to your diet, connected to your activity, and connect, for example, you have if you're burning fat, you have change in acetyl-CoA. This can change your acetylation. This can change the level of acetyl-CoA, and so on. You have change in acetylation or within the mitochondria. So it's something which is it's not an enzyme that mediate the effect. It's environment that control the effect. But we don't know yet whether in a long-lived animal, whether there is change in the diet that is important for this effect on acetyl-CoA.
It can be also metabolism, correct? Because metabolism.
Right. When I say diet, I mean metabolism.
Okay, yeah, that's interesting. Another interesting phenomena that very related to longevity, in my opinion at least, is some of the DNA repair pathway were also highlighted in your finding. Can you elaborate on that?
Okay, so I will go now to Peto Paradox, which I'm sure that the audience is familiar with. But the Peto Paradox is what you mentioned before. We have a very simple rule in longevity. The bigger you are between species, not within species, you live longer. Elephant, we live longer than, let's say, mouse. Whales live longer. Humans live longer. And so on. I will speak later about the cat family, which is a unique example of this. But in general, the bigger you are, you live longer. And when we research on specific acetylation sites that created in long-lived animals, we found that many of them are related to or happen on two different set of protein. One is DNA repair, any type of DNA repair. The second is cell cycle. Now, why I'm saying it? Because there is a paradox that's called the Peto Paradox. The Peto Paradox asks the following question: how come that if you are bigger, you live longer? If you have many more cells and you have many more years, so you have much more opportunity to develop cancer. Because let's say an elephant has millions of cells, million times more cells than a mouse, and still don't die from cancer. So they need to find a way to escape the fact that they live longer, they have larger body size. One of the ways to do it is to find ways that you block cancer, because you must block cancer. The fact that we found that many of the acetylation sites happened on DNA repair and on cell cycle, at least for us, it reminds us the Peto Paradox. So we thought that maybe because of the development of this acetylation site, longer lived animals, in other words, larger animals, found a way to block cancer. So what we did next, we focused on one site. It's called USP10, which is involved in DNA repair and in cell cycle. And we found, and USP10, one of the things that it's doing, it's regulating cancer-specific markers like PCNA. And we found that if you have higher activity of USP10, you have much higher level of PCNA. And we did it in a big data experiment. We showed in at least 3 different types of cancer. What we did next, we took the human protein and we converted it, or we changed it to the mouse protein. Again, only this site of acetylation, we changed 1 amino acid. And we found that the level, that once we did it, the activity, which is the pro-cancer activity, becomes 3 times higher. Meaning that if we are mouse or small animals, when we don't have this acetylation, now we develop cancer. When we're doing evolution, we acquire this acetylation, we block cancer. This allows large animal to live longer and basically to solve the Peto Paradox. I hope that I was clear enough.
Yeah. So basically, you took a mice fibroblasts or something like that, and made them transgenic, and then check the effect of tumorigenesis.
Even simple as that. We just took the mouse cells, we switched it to human, and we followed the level of PCNA. Okay. This was the first step that we did. This was enough. We showed that now we have 3 times level PCNA, which is a significant marker of cancer.
Got it. So you just look at the expression of that.
Of the target.
Now, interesting point about cancer. Again, I remember it from the discussion with Vera Gordonova, and also you mentioned it in your paper, is that in long-lived animal, example is the elephant, they have, I think, 17 times more p53, which is one of the most important tumor suppressor than us even. And my question, and you mentioned before also that, and it's true, p53 is acetylated. Have you looked at that specifically in this study or not, to see whether there are changes in the acetylation location of p53 between long-lived and short-lived?
We didn't find anything about acetylation specific acetylation of p53. I think p53 is one of the most conserved, or let's say a very fundamental protein. So evolution do not play with p53. Okay. And one way to play it is like to have 40 times higher number of p53. But in the sequence, you see very few changes. Let's say post-translational modification of p53, it's conserved from mouse to human. I think almost every site is conserved. We didn't see anything at p53. Also, I think you want all these changes that acetylation allow you, or any PTM allow you. You want something which is subtle. You don't want something which is 1 and 0, because if you have something which is severe effect, I don't think it's allow us to respond well to change in environment as something which is subtle.
Yeah, and just for the audience, for everyone that doesn't know what is p53, p53 is maybe the most important tumor suppressor gene in our body. Around 50% of the cancer that we accumulate have mostly a point mutation in p53 that basically allow the tumor to grow and to flourish. And there are a lot of labs that study p53 and a lot of drugs that are trying to be built against that. And it's a very fascinating domain, and whoever will be able to crack the code there will definitely make a big change in our response to cancer. Another interesting point, Haim, that you raised is about cat. You said that cats are a bit different. So can you elaborate about that?
I just mentioned it as we spoke, because in cats, and I don't know yet why, all of them have the same lifespan. Whether if you are a lion, or you're a leopard, or you're just the cat that I have in my back garden, they all live very similar. You don't see changes in cats. So this change, this correlation between size and longevity, I don't see it between cats, and I don't know why. This is one of the projects that we want to look within PTM, why you don't see the chances between lions, which are 10 times at least, I don't know how many, 50 times bigger than a home cat. But
Yeah.
it's the same.
Yeah, interesting, very interesting. I didn't know that. That's definitely fascinating. So either you have a small cat in your house or a lion in your house, they will live the same,
For the same. If
the
you
same
choose the
lifestyle.
animal, if you choose a pet and you base it on longevity, it doesn't matter if you take a lion
Yeah,
or a
yeah.
cat.
Yeah, yeah. So, so that's a very interesting paper. And do you see any possibility to design, I don't know, small molecules or a peptide or any other, I don't know, genetic aberration benefit of dose acetylation state in human, namely? Do you see a therapeutic implication of your discoveries?
Okay, yes. So I will say a few directions that we are looking at now, and we're trying to explore them. Let's start from what for me is the most exciting one, and this is the following. So human are arrogant. We need to think, we always need to remember, when you think about evolution, we always believe that evolution was done and we are at the end of evolution, right? Everything was done just for us. We are the best creature of evolution. But actually, in longevity, we live 100 years and whales live 100 years, right? And they are very far from each other in regard to evolution. So what we're doing now, we search for specific PTMs or changes in PTMs that happen in long-lived animal during evolution, but didn't happen yet in human. Meaning that all we need to, and we have already found a bunch of them, means that all we need to do now, we need to take this site and let's say be faster than evolution and do it and take it back to human, to take the sites that happen in whales or in primates that didn't happen yet in human and convert them or translate it into human to see whether we can use them in order to extend longevity. This is a long range.
So basically what you are saying, expedite the evolution by learning from a creature that you believe that are more advanced in evolution than us. And Haim, PTM is post-translation modification, correct? Just for
right.
Yeah, okay.
No, I'm not sure they're more advanced than us. They say we are arrogant, but definitely they live like us.
Maybe right. They are. Yeah.
They live like us. And they live, even some of them live more than us. So they found a way to survive longer than we found.
Yeah.
So this is one way that we're looking now. We create a database of this so you can focus on this side, see whether or not you can change longevity of even long-lived animal. The other approaches are either to search for specific disease that have variation on this side. And we're also doing this. So we're searching for variation within the population, within the site, which are important for longevity, and to correlate this to specific phenotype. So now we can fix it back by reverse it to the common variation in humans. So this is one way, this is another. But you can also put aside the modification. So we always, in the last few months, we think in the lab whether we should target exactly the modification or the protein. And I think it's more easier to target the protein rather to target the modification. Because if you want to target the modification, let's say you have a protein X, and you know that some modification may improve its activity by 20%.
Yeah.
And you know this improvement lead to extreme longevity. One way is to struggle and to express, modify the protein, or to find a way to connect that acetyltransferase to the protein in a specific environment, and by this change the modification. This is difficult. Yeah. More easy is to find a way. So now you have a target of longevity, and you can find drugs that will activate this protein by 20%. You don't need to. This is much easier, I think, in terms of biotech company to do rather than to target the acetylation site directly. This is way more difficult. Yeah. We're trying to do it, but it's easy if you're using a mouse. In humans, it's a bit difficult. But to target the protein and specific, since we have many existing drugs, all we need to find is something that already targets this protein. We can take this drug and use it for something else.
Yeah, makes sense. One follow-up question coming back to the beginning of our discussion about the 36 and the difference between males and females. Have you looked at that in acetylation or post-translational modification? Are there some difference like that?
Okay, so we did, it's not published yet, so we did the set
Okay.
of 36 dependent acetylome, but unfortunately we did it only in males. This is a question of budget. You know, you need to grow it to
Yeah.
raise old enough mice, and we do it only in males. We didn't do it. It's an excellent idea, but we need to do acetylome of females and in males. We did it on the acetylome of 36 only in males, not in females yet.
And that is more like a literally post-translation modification because our genome is the same other than the Y chromosome, correct? So it should be, the site should be the same, but maybe the acetylation might be different because of, I don't know, some post-translation modification.
Right. They have also another X chromosome, but most of it is silent.
Exactly. X chromosome is very limited, has a very limited information. Very fascinating discussion, Chaim. I just want to, I know that both of us are very excited, but what do you see are the limitation of this paper that you published, just to be sure that we are right? What are the limitations in this study?
Okay. So first, I will say one comment. I don't know how many, so do you. We are in the science business for 30 years, 25, 30 years. I never met one scientist that believes there is limitation in his study. This is something that does not exist, but always we ask about it, right?
Yeah.
So it's like not existed, but I think that as it started, as you mentioned in the beginning, what we found is association. We didn't find direct link. So it will take us, that's exactly what we're doing now. We try to find, let's say, the top 5 sites, and to prove it in vivo that by manipulating this site, we can change the lifespan of the animal. Since we are very ambitious, we want to have 50%, even double the lifespan of mice, because we want to take it even farther than the 30% that we did before. Yeah. But definitely the limitation is that we start from computational approach, and we have very important list, and we have a very nice list that make sense that it's related to longevity. Most of it is longevity associated proteins, so we know we're in the right direction, but the next step will be to take it and at least in 1 or 2, that's exactly what we're doing now, site to really extend the lifespan of a mouse.
Yeah, no, that's very exciting. And if you try to compare what you describe in this paper about, let's say, global acetylation versus the effect of 36 or caloric restriction or I know rapamycin, what is, I know that you don't know, but what do you think about that? Would that will be a longer effect, a better effect, a less effect, the same effect, or it's hard to say?
Okay. First, it's hard to say because I would say it's hard to say because if we're taking back to short-lived animals, definitely that it's not enough. I don't think that we can take the lifespan and change it to human lifespan, like take 2-year-old mice and now make it 70 years old. It's not the limitation of the physiology of the mouse.
Yeah, yeah.
But if I think about the effect, I think the effect will be bigger because we are not, when you speak about rapamycin, again, you're looking in one pathway, mainly metabolism. I know that rapamycin also, mTOR, has some side effect, but probably has some side effect, but you focus on metabolism. Here, because we did a unbiased search, we touch many longevity-associated pathways. So the effect is across different longevity-associated pathways, like metabolism, DNA repair, senescence, nutrient sensing, and so on. So you have something which is bigger than the single pathway that you focus on. Yet, I don't think that we can take a mouse and make it live like a human. But definitely the effect will be more than 30%.
Yeah, the question is also, to make a mouse live longer is very exciting, I'm sure, for scientists like yourself, because, but the other question is, what is the implication for human? Because as you said, we are very arrogant, but still we are on the top of the lifespan. So how can we apply it to human, because what you're saying, okay, we learn from human how to make mice live longer. I'm not sure that our audience care too much about
Right,
mice living
right.
longer. So what, and I know that it's a tough question, but what is your argument about that? How can
Yeah.
that help us to live longer?
Well, I'm sure that it can help us to live longer. First, because as I said before, we can see what's happening in other branches of evolution and translate it to us. Second, we can find pathways and protein which are important for longevity.
Yeah.
And if we did the rate, or we did the curve of change in acetylation during evolution, it's not that it happened one day and immediately all of us live longer, or all the animals live longer. It's something very gradual during evolution.
Yeah.
It means that we can still have, and it's not come to a plateau.
Yeah.
Meaning that we still have a space to improve human longevity.
Yeah.
So what we can do now, we can focus on this side, and especially because it's on and off. Let's say the on is important, we can push it toward the on or toward the positive effect, and by this extend human lifespan.
Yeah. Yeah. No, makes sense. And I think that the point that you said about a screening versus what a lot of labs are doing today, that looking at rapamycin and metformin and I don't know, even 36, that's something that actually is standing under the lamp. So we are looking at it because we know about it. But in my opinion, and it sounds like you agree with me, the revolution will come from something new that we don't even know about it. So doing screens like that, they are very risky, but in other way, as we know, risk reward is higher. Very risky, the reward might be much higher. So we might find something that allow us mice to live to 5 or 6 or 10 years. As you said, they cannot live 80. But we haven't seen such an effect in mice. Maybe the highest one that I remember is around 50% or something like that, maybe caloric restriction. But we don't see more than that. So I agree with you that doing screen like that, even so that they are very risky, they might provide to us a very high reward. Do you agree with that?
Definitely. More than that, it's something which is hidden in our paper. We did the same screen. See, when we used 2 different databases. One was maximal lifespan, the second was median lifespan. We wonder whether it be the same pathways that affect your median lifespan are the same pathways that affect your maximum lifespan.
Yeah.
And I can't, and I'm sorry that I don't remember exactly what they are, but it's not exactly the same pathways.
Yeah.
Like 90% of the pathways that affect your maximum lifespan also positively affect median lifespan. But there's still a small group of pathways that affect median lifespan.
Yeah.
And this definitely have an enormous effect on humanity.
Yeah.
And also there are pathways that affect your maximum lifespan. So we can get, and as you say, we look on something which is unbiased, and we find pathways that we even didn't think about it before. And now when you focus on it, you might be able to translate it into human longevity.
Yeah, no, that's great. So, Chaim, congrats for the very exciting paper that you published recently. I'm really excited about that, and it was a pleasure reading it and pleasure talking with you about that. One last question that I always ask our audience, our expert that I interview, is what is your best applicable tip for longevity that you would like to provide to our audience?
And you asked me that 2 weeks after the taurine paper was published, right?
Yeah.
Right? Like 2 weeks ago, we can speak about take taurine and live for longer. And I'm sure you read the couple papers that basically say it's not the right supplement. At least it's not a good marker.
Yeah.
So the only advice that I have now, it's very, I try to be very, not to say something out just like that. I think the best advice that is to align, and this is something that people can do, to align your activity cycle to your diet cycle. This, I think, it's a good advice, meaning that when you're active, or in our term, during the day, in mouse, of course, it's the opposite. Eat when you're active, and during the night, do your fasting period. I think this is advice that I can give to be sure that it works also in human.
Okay. So basically, if I'm trying to make it applicable for us a bit more, is don't eat too close to bedtime, and try to eat as early as possible the last meal, not to interfere with your sleep. That's what you're saying?
Right, not during the night. I know it's a very simple grandmother advice, but it still works.
Yeah. I hope that none of our audience is eating at night when they're waking up to go to the loo. But yeah, it's definitely a great advice. Thank you so much for that. And we are coming up on our time. Thank you so much, Dr. Cohen, for being here. Looking forward to exploring the research in the field of longevity each week with you and the other leading scientists. For more information, please go to www.instatracker.com/podcast. Thank you so much, Haim.
Thank you very much.
Thanks for listening to Longevity by Design. Please subscribe to this podcast on Apple, Spotify, or YouTube. Longevity by Design is powered by Inside Tracker, a personalized health optimization platform that helps people improve their lives by improving their bodies from the inside out using personalized, science-backed recommendations for nutrition, supplements, and lifestyle changes. To learn more, visit insidetracker.com/podcast.